Students, today we are going to learn an experiment which is based on the physical process known as osmosis. What are the things we are using for this purpose? We are getting hold of a large potato, knife, 10 to 20% sugar solution, petri dishes, beaker, water and pins. So, how do we start with it? All of us have seen a potato, correct? All of us are very much acquainted with it. Now, this is a peeler. With the help of the peeler, we are going to peel out the skin of the potato. It is a very easy process which you might have done in your kitchen along with your mummy. Now, it is peeled up. Okay. Then with the help of the knife, you are going to cut it into half. Now, this is a half peeled potato. We have to see to it that it should, this is a petri dish. We are going to make it stand here. So, let us cut it down to make the base flat, right. One more thing we need to do, we have to dig out a well. We have to dig out a well from inner core of this half potato. With the help of the peeler and the knife, or if you have a scoop at home, you can dig out a well. Be very careful when you dig out a well, because it should not become so thin that hardly any cellular material is left at the base. It should not become very deep, neither too shallow, right. It will appear like this. It has already been done before, and if we can see this potato, I hope it is clear to you, students. Now, inside this potato you can see one pin has already been marked, and what is present here? We have put in water. This is my control for this experiment. Control for this experiment, and this petri dish contains plain distilled water. Similarly, in another potato setup B, what have I done? I have cut out a well, but here I have put in a solution which has already been made with the help of sucrose, sugar, and a portion of water. This solution had been kept in this well in setup B. We will leave this aside for about more than half an hour. After we come back, we will just notice any change in the water level or the solution level. Thereby, when we find out in comparison to the, I had done it an hour before. So, here the pin which exists shows me the initial level of the solution, but when I came back after an hour, is it visible to you? The water level inside the well has increased, and I am putting the final pin here. I am putting in the final pin here, which is on top of the initial one, whereas in my control experiment, this was the initial level. This was the initial level, and what do I see? Now, the level remains more or less same. I am putting the pin on the other side of the well, and you both can compare the initial and final levels. It more or less remains unaltered. So, definitely something has happened in set B setup. What can happen? And when can the external water solution enter into this well correctly? And one more observation, students, mark it when you touch the potato. How do you feel? Is it softer or tougher? Why do we need it? Let us understand through the explanation. So, here we see the potato has been cut open, is not it? This is the initial level as shown, and after we came back after an hour, in set B, this was the condition. The final level shows that it is higher up than the initial level. Now, we are trying to understand this case. This can only be happen when the solution from outside water will enter through these living cells into this well. When will this happen? Why will this happen? Let us understand osmosis for that purpose. This is a semi-permeable membrane represented by the dotted line. Blue circles, they represent the number of solvent molecules and the purple ones are solute molecules. So, if we think about two regions here, what can we see? The concentration of the solvent, blue colored circles are more on this side and lesser on this side. So, the solvent molecules will have a tendency to move from this area to this area, where they are more to where they are less. Question comes, how long will they move? They will move until they are equal on both the sides. As they are moving through a semi-permeable membrane, remember always this is known as osmosis. Unlike diffusion, there is no involvement of any kind of membrane, right. So, here also this water molecules, and here initially we had put in sugar solution. Sugar solution is a bit concentrated, it has got certain solutes, solvent particles are less, but here water is only having solvent particles. From here they are moving through these cells, living cells into this well, thereby increasing the level of solution inside the well. This is known as endosmosis. From outside, solvent molecules are travelling inside. So, this is endosmosis, which has resulted in increase of the level of solution. So, we call this a potato osmoscope. One small interesting fact, in place of a raw potato, if I use a boiled potato, what is the difference? It is no more living, is not it? Because when I boil it, the living cells, they die up, and so I had performed the same setup with this boiled one, but I did not get any result. So, this is the reason, because the living cells are not involved.